SRL Diagnostics.
Hello, I am Dr. Anupkumar Rawool, Associate Director, Department of Medical Genetics, SRL Diagnostics Lab, Mumbai. Today we will be discussing about certain genetic tests, what we call as exome sequencing. So everything revolves around next generation sequencing. So we need to keep in mind that there is no single genetic test which will give you information about the entire genome. But one test that can give you maximum information is whole genome sequencing. But again, only 1% of the human genome is protein coding or functional, what we call. And testing that particular region becomes more relevant because 85% of the abnormalities lie in this region, what we call as exome or the whole exome. And there are about 19,000 genes which are hosted in this functionally important region of the DNA, the whole exome. So when it comes to testing, if you have certain genetic disorder where the clinical picture is clear and you know what are the genes that are responsible, it is always good to go for a focused test or certain specific genes which need to be sequenced, what we call as NGS panels. Then there is a test what we know as clinical exome. So what is the difference between the NGS panel, clinical exome and whole exome? So if I have a clinical picture of say a hemolytic anemia or a bone marrow failure syndrome, so then I am going to look at those particular set of genes which are relevant to this clinical phenotype. So I would prefer an NGS panel. So what is the importance or advantages of doing an NGS panel test? Here, a small number of genes are looked into or interrogated or investigated at a higher depth, more coverage, and chances of finding or picking up any abnormality in that particular focused region is very high. So if clinical picture is clear and underlying genes are known, it is always better to go for a focused test like a NGS panel. Then what is clinical exome sequencing? So there are certain labs which have come up with this concept that they have clubbed all the genes of clinically important disorders in one panel. So at SRL, we are covering more than 6,000 genes in clinical exome panel. So the idea is if there is a genetic disorder of which the clinical picture is somewhat relatable to specific known conditions, you can figure out the differentials. So you need to interact with the lab that what are the clinical differentials you are looking at, what could be the underlying genetic abnormalities, and are those genes covered in the said clinical exome panel. So that is clinical exome. When a picture is not very clear and you want to look at the differentials in one go, but you need to get in touch with the lab so that the clinical details, the phenotype details are shared, and at the same time you can interact with what are the genes related to that phenotype that are going to be covered. Next is the whole exome. So whole exome, more than 19,000 functional genes are covered. So particularly in those cases where clinical picture is not relatable to any known phenotype or genetic disorder, or there is a huge overlap, in those situations whole exome sequencing can be done. And also it helps to pick up rare conditions, novel abnormalities which otherwise may be missed on a next generation sequencing gene panel or a clinical exome. Thank you.
SRL Diagnostics.